Hello, Bradley.
Hello, Lalith. How are you?
Good, good. It's been a
Great.
long time.
Yeah, it's been a long time. So, what have you been up to, Lalith?
So, so I've been working on that work, and so Richard have been, you know, you know, he's kind of sent our, like a paper uh, to one of the editors in biosystems. So he kind of reached out to us, and he's suggesting that there is a deadline timeline around like November 1st week to submit the work. So I was preparing the manuscript according to that. So I just wanted to give a brief out about that work. So what we're gonna do about it, and a short presentation where I can, detail what has been done, and what the things that should be included, and take a review from you all, and maybe you can add those concerns into that manuscript. So,
Yeah.
so I thought,
Yeah, that'd be great. Yeah, it sounds like it's coming along. So, yeah.
yeah,
So, yeah, do you want to present today or?
yeah, I can
Okay.
present now.
Yeah, it's
just
good.
give me a
All right.
Yeah.
Hello, Susan and Varshith and Morgan.
Yeah.
Okay.
So, is it visible?
Yes.
So, yeah. So this is the work, and it's start. So the problem statement of the work, what we started with one of this Google Summer of Code project in 2025. So it was meant to understand how this complex multicellular structures of like C. elegans during embryogenesis, how do they evolve, and so as taking as an inspiration from the previous works of Bradley, that the anastomosis can be seen in the during the embryogenesis of the C. elegans. So not just fission it does, but also there are like certain fusion interactions that can be handled, and we can also see certain synthetium cells which are responsible to form tissue level organizations and all. So we were very intrigued by understanding what could be seen at a computational level. So we call this as like a group or higher order interaction, group level interactions. So because general standard graphs has a single pairwise interaction, whereas hypergraphs has like a higher order interactions. So we have a cell to cell interaction, such as a parent, it's been divided into like two daughter cells, but they do form a small community where there is a group level interactions. In order to see that, so what we do is, so we have the data which is represented into two certain manner. So one is the spatial proximity or the spatial relationships, where we have the XYZ coordinates of where exactly the parent cell has started, and how it was divided. And we have the lineage information, where from which parent to which daughter, and from that daughter acts as like a parent to the another, like while during the fission, and how it grows. So this higher order interactions we wanted to model with hypergraphs. And we wanted to study this evolution process of embryogenesis, and see how over time this evolution happens. So in short, so we develop a dynamic hypergraph neural network which integrates both spatial and lineage interactions to understand and observe and predict the behavior of C. elegans embryogenesis. So this is the main idea of like the workflow of the way which we see. So here we have a single cellular organism, and later it divides into multicellular and it becomes an adult one. So during this, we have a single cell, and at all the time steps, we have like the way in which it has been divided. So, and also this information, including the lineage information that these the entire the temporal So we model this as a graph, and using that graph, we take the lineage and temporal information and then feed it to the hypergraph neural network. And this network will help us understand that what kind of associations it actually forms to make it more reliable and more correct predictions. Suppose we see here, initially it starts with understanding that, these three do form a certain hyperedge because they have a certain type of associations. So this might be because of they form a same, they belong to all the same tissue cells, or from which a single syncytium has been started, and so it evolved. So this is the pipeline that we have observed. I mean, like, so drawing these hyper edges on top of that is the main challenge here. I mean, like, how do we say that these edges are correct? So we design a complete neural network workflow, which actually does that. Tries to understand and optimize and capture the best higher order interactions in this embryogenesis of C. elegans. So this pipeline model is basically, we call it as dynamic hypergraph neural network, and at each time step we have a 3D point cloud, or like a 3D point cell, and over the spatial and the bird times. And so we build two kinds of hyper edges. So we don't just rely on spatial hyper edges by finding the proximity, but also we have the lineage information, and we see how these connected sibling cells, the parent and daughter relationship, and how they have been acquired. So we formulated it in a way, and going to that, so we then combine this into a common hypergraph with both spatial and lineage relationships, and this we give it to a neural network. So we have here hypergraph neural network. So this tries to find out the associations, and we have a neural network, a layer which is called gated recurrent units, which tries to understand how this time temporal during the time evolution, which time step is actually trying to acquire. So the temporal features have been acquired by this layer, and the spatial features have been acquired by the hypergraph. So this mixture altogether will give us a better associations in the how, I mean, like, which can be grouped together, which cells can be grouped together. So this is the final projection head, which gives an embedding layer. So based on this, we acquire the features from like neural network, understanding the both spatial and lineage information. This layer has all the features regarding the whole time series and the information that it has. So based on that, we actually use these features as like a lower level representations, and this help us to map onto uh, the data that we have, so that it will help us to understand. So we have a visualization that probably Varshith can show. Uh, uh, so we actually ran the experiments. So when we ran the experiments, we were able to get the complete embedding space, how the neural networks tries to understand this whole embryogenesis process. So this I did it in the GSOC project, and Varshith was able to reproduce it, and he'll show that. So I think this is a representation space. Now, when we try to point out at a certain embedding, any embedding, so we will be able to see that what it actually is. So that is the lineage information, and the XYZ coordinates and the cluster information that it associates with. So from this, we will be able to find grouped clusters. with a certain behavior, and then from which we will be able to get a good understanding of okay, which type of cells have been come together, which have been drifted apart, and which are actually forming associations and which are forming clusters. So we'll be able to see that. So this is just the last time step embedding. So we can use all the time step embeddings. I mean, like when it started and when it ended. At every time we can map these embeddings and see like how are they grouping, how are they associating. So yeah, I'll get back to the presentation now.
Okay. So this is UMAP. This is like a two-dimensional UMAP that you're producing off of the
Yes.
basically five-dimensional data, and it's that
Yes.
space. So how do you interpret that space? I'm curious.
So there are so whatever the information that we acquire from a neural network, so we use that neural network to embed it into a certain space, so that it actually has the entire representations of the five-dimensional information into that small space, so that we will be able to say that each data point that we have in the UMAP embedding associates with the relationship that each and every cell has. Suppose they come grouped together, then we can say that okay, these types of cells have a similar behavior, and this type of cells might have certain characteristics. So that is the key challenge that we are trying to understand here. So
Yeah, the reason I ask that is because it's very popular to use this in like genomics and that, and people will misinterpret it and say that they're clusters. I mean, but they're not technically clusters, but they do have this spatial, or they do have this relationship where if they're together in space, in the XY space, then they have something there,
some
So, I mean, yeah,
meaningful,
it's a subtle relationship, and I don't think anyone really explains it. You know what I mean? Like when you read papers.
Yes.
Yeah.
Yeah. So yeah, we were trying to understand the similar, because this was a last time step embedding. When we're trying to plot it in a first time step, we have only single cell. So we are trying to understand that okay, in a single, if there is during the evolution, at least for like 10 time steps, we'll be able to see that two cells have come together. Then next, did the two cells which were came together in the first time step, are they drifting apart in the next part? I mean, like during the next successive time periods, are they drifting apart? If they're drifting apart, what does that mean? So we are trying to understand that part. So I what I and Varshith discussed was it will be more meaningful if we actually not just visualize them, but we make an interactive tool and make it online, so that when we click it, then we'll be able to see how the changes are happening, so that people might like suppose we have a certain perspective. If two to three different people see that, and they might have a different perspective, so from that visualizations, maybe we can draw some good conclusions, is what we believe. So that's going to be in our next step. So
Yeah.
yeah. So I'll carry on with the presentation now with yeah. So this so what we are trying to do is right now is we have our model, which is the temporal block is the GRU, so which is gated recurrent unit, and we have our hypergraph convolution, so which is like the spatial, and we don't have a transformer model as of now. So we have the default configurations that is actually working from the paper. So this is which it is done in the first. So what we thought was we can mix with certain base models, and we can mix with different types of temporal blocks, and we can use attention layers in the hypergraphs and all. So we can do a good baseline runs, and from that we can able to test it. And second, we thought that it would be great if we actually include the interactive visualization tool. So that is the second. And third part is we are still trying to plot visualizations in 3D and 2D, and we are able to bring some good interpretations out of it. So this is ongoing, and so if you find an update, then probably we'll mail it, and so if you can give us a review on that, it would be great.
Review on what?
A review on the manuscript that we are trying to submit.
Yeah, but like, what is the action item? Like, what would you need?
I'm sorry.
What would you need for this? Like in terms of a review, you said.
So in terms of review, I would, I would appreciate if there is a, thorough understanding of whether the interpretation, as you have mentioned. So we are trying to interpret something out of it. If it is, it might be constricted to a certain perspective, whether it can be broadened or whether more biological insights that could be added into it to make it more enhanced. So I'm not really from like a deep biology background, so I would appreciate certain things into that.
Right. Oh yeah, that's definitely the case. Yeah, I think probably the best way to go about this is to have like a, write up some of these results, and then if you send me like a copy of it, I can figure out where I can contribute a review or some insight and put it in, and then that'll be I think like it looks solid. The work looks solid, and I think the best way to really kind of go about this, especially for like biosystems, is to have visualizations, have a lot of maybe about 6 or 7 graphs where and there's an economy of graphs where you want to get like the most descriptive graph possible. So one might be the first one you showed, where you show the process of going from the embryo down to the hyper networks, and then kind of explain in the paper what that transformation is, why it's important, and then the next one being maybe like the embedding space with
Right.
UMAP, and then say why that's important, and then maybe have a few more results. I like the table you had at the end, and I kind of wanted to go back to that
Okay.
a bit. Can you go back to
Okay.
that slide?
Sure.
Okay, yeah. So this is like this is like all the different models that you can kind of have.
Yes.
Okay. And this is hyperedge convolution temporal block, just variations on different technical things. And then
Yes.
notes. So do you have like a metric for this, like where you're testing, like benchmarking the different methods?
Yeah. I have all the evaluation metrics, and I'm trying to you do it in a standard the machine learning pipeline, like how we do it in that. Following the similar protocol, like 80% of the data to testing, 22 or 80% data for training, 20 for testing, and see like how the predictions are well close with the the actual ground truths, because we have the entire label data, so I have made it in.
So yeah, that's good. That would be good to have the table, and then some of the maybe one statistic. You don't want to overload, because this is not a machine learning journal you're targeting.
Yes.
And then is there a way to get like, I think that the final graph would be something like taking these different methods and showing some output from them. So it could be like the UMAP visualization for the different methods, and then like either having panels where people can visually compare them, or like I don't know, overlaying them is a little messy because there's going to be a lot of overlap, I imagine.
Yes, I can have different panels which can visualize them. Yes.
Yeah, that's great. And then this is something where we've kind of have this I've had some presentations where I give a talk about like the different levels of dimensionality in kind of our approach is like to take the three-dimensional, like you get the three-dimensional data, the XYZ data from cell tracking, and then you can add in other dimensions. So like you have all sorts of different dimensions. You can add in time, you can add in angular data, you can add in cell state, you can add in all sorts of things, and then it makes the model more and more higher and higher dimensional. So like, you could have a 5-dimensional model where you have 3 dimensions of space from your cell tracking, 1 dimension of time from your cell tracking, and then sort of derivative variables that you get from analyzing the data. And then you have like this 8-dimensional space. And then that 8-dimensional space, instead of the point cloud, which is 3-dimensional, you have this 8-dimensional space, which you can then reduce to, using some sort of dimensionality reduction, or you can, do things like do time series, like what Jayadratha did this summer, all sorts of things. So I wanted to kind of put that in the paper too, like kind of that formalism for that and discussion, because we do this and we have other papers where we do this, and so I just want to have a continuation of that. And then having this, I think it's nice to have the hypergraphs approach where you have like basically a statement of we can take things from a lineage tree and then turn it into this sort of representation where you can recombine the nodes into different categories. The only, well, one thing maybe we could see is more actual hypergraph sort of representations. So, like, do you have a good method for showing the hypergraphs as like these, uh, like showing the nodes within another hypernode and then showing the hyper edges? Because that's going to be very hard for people to understand and visualize.
There are few type of visualizations where I tried it in 2D, but in 3D, I think I came up with an approach which is actually feasible. This is called star method. So the star method, what it does is the interactions, it will place it as a small point, and from that point, whatever the nodes are connected, hyper nodes, so it will be connected. So it's like it doesn't look like we are drawing an edge on top, hyper edge on top of all these things, but it look like the way the associations have been formed. I can do that. I mean,
Okay. So it doesn't look like the nested groups that I mean, the nested groups are nice. But what does it look like? Could you describe it a little bit, or?
I think I have plotted it in long while. Like I don't have right now, but I can.
Oh, okay.
I can show you. It was long back. I have to. So maybe in the next week meeting, I'll show how it was there. I mean, so it looks more like a cluster, and there is a point which actually, from which that point, all these points are connected to that cluster, saying that these two belong to the same. This is a hyper edge which is actually connecting all these points. So I mean, like all the points, suppose let's say hypernodes are, I mean, like in circular color, we put it as a square box and say that this square box is the point. I mean, like this is the association, the hyper edge which brings all these things together. It carries all the information into this. So we can, I mean, like, it's not very intuitive as drawing a top line which is on top of that, but it's more like we can do it in a different fashion. But yeah.
Okay.
In Python, visualizations are quite little hard.
Yeah, I know. It's like there are specific visualization tools that usually do a lot better. But no, this is great. This is great. And then so that's the idea for the paper, like really kind of having want to be accessible to the biosystems audience. So it's just a matter of figuring out how that looks. And I think visualizations are the key. Not having a lot of statistics without interpretation or like methods without interpretation. Yeah, well, the best way to do this is just to kind of go through and with those things in mind, and then we'll review it and see where it might be a little
Yes,
hard
yes,
to understand. Because
yes,
it's very hard for people to wrap their heads around it if they're not used to the,
yes, yes,
yeah.
yes, yes. So it should be more convincing and easily understandable too. So rather than high level technical details have to be, should be meaningful, that we are trying to make it more to, applying it to a place where this is more like an applied topic. We are applying the hypergraphs into the embryogenesis. It should, I mean, like, the writing should actually make more sense about, like, how biologically this is meaningful. So
Yeah. Yeah.
so that consumes a little time to do that.
Yeah. That's good. All right. Did you have
Thank you.
anything else, or?
I mean, like, I've been meeting monthly once or twice with Richard Gordon, and I and Varshith both are like meeting, and we are we have submitted a paper where it was halfway done by Richard on the exponential of the exponential neuron. So there are like this. There are number of cell types that are increasing from like long time, and so during the evolution, these neurons have been seen as, like, when we try to plot it in an logarithmic graph, it's exponential. The number of cells are increasing exponential in a logarithmic scale. Yeah. So that's one of the interesting thing that we have observed. And so, yeah, the hypergraphs and this.
All right. That sounds great. Thank you.
Yeah. Thank you.
Yeah, that's great. So yeah, Lalith, congratulations on making some headway on this, and having the enthusiasm for continuing GSOC stuff. Yeah, so I'm gonna be giving a lecture to the OpenWorm Foundation next month on like the yearly report on DevoWorm. So I'm gonna include some of this into it.
Sure.
All right.
That would be great.
Yeah. So this is a paper where, let's see. Okay, so this is a paper that was published last year, and this was in Nature Structure and Molecular Biology. And the title of this is Precise and Scalable Self-Organization in Mammalian Pseudo-Embryos. So this is an interesting topic about like creating embryo models for analysis. So a lot of times it's hard to work with mammalian embryos. And we're interested in a lot of the properties of those embryos, but we can't, for maybe sometimes ethical reasons or a lot of times technical reasons, work with actual embryos. And so, if we're working with C. elegans, it's actually fairly easy to work with the embryos, work with something like zebrafish or Xenopus, which is the frog. And it's, we can do experiments there. And that's why they were model organisms, because they're very easy to work with in terms of the embryo. But in mammalian embryos, it gets a bit harder to do certain things in those models. And so this is where they're actually presenting this model of a gastruloid, which they're calling pseudo-embryos. I think this work, people were calling them virtual embryos for a while, but that's kind of like implies like computer simulations. So that's, oh yeah, I got to share my screen. I got knocked offline here. All right, so this is the paper from Nature Structural Molecular Biology. Okay. So this is the paper from Nature Structural Molecular Biology. This is Precise and Scalable Self-Organization in Mammalian Pseudo-Embryos. And the authors are here, and this includes Thomas Greger, who sort of leads this group in this research. So I'll read the abstract. Gene expression is inherently noisy, posing a challenge to understanding how precise and reproducible patterns of gene expression emerge in mammals. So again, we can study gene expression in like Drosophila embryos. This is where we got a lot of information about sort of morphogenesis and the sort of collinearity of Hough genes and how those are expressed in the embryo. So we have some good model systems for looking at gene expression in embryogenesis. Um, but it's harder to do in mammalian embryos, and it's very noisy as a process. And so you want to minimize that noise as much as possible. So what they're trying to do is minimize the noisiness of gene expression. So oftentimes you get what they call biological noise, and then they call technical noise. Biological noise is the noise inherent in gene expression. So genes are expressed usually when a cell is stimulated or an environment changes so that the expression pattern, the gene is upregulated or downregulated. So if you change the rearing temperature, or if you change its exposure to light, or if it kind of experiences some chemical signaling from other cells or from the environment, the gene in question will either it'll either rise in terms of how it's being transcribed, or it will be quieted in terms of how it's transcribed. And so, without getting into too much technical detail, this is something that involves the regulatory machinery of the gene. It involves upstream promoters, enhancers, silencers, things like that. It involves the opening and closing of chromatin and all of that. So there are a lot of sources for noise there. And so you get this noisiness in terms of the biological expression. You also get biological expression when, during development. So you go through different stages of development. Sometimes genes are turned on, sometimes they're turned off at different phases of development. And if you go to something like cell cycle, the switching between upregulation and downregulation of genes is very precise, and it has to be very precise in order for that process to work. And so that's biological noise. And then technical noise is where you get noisiness in gene expression, not because of the biology, but because of the technical methods you're using. So if you have something that you're working with in culture and you put the cells under a lot of stress, they will express certain genes, or most genes, at a level that is not characteristic of their biological background. In other words, if I have a cell culture and I have it under stressful conditions, if I raise it in a high heat environment, or if they don't get enough nutrients in cell culture, then they will express genes. There are all sorts of stress pathways in the cell that turn on genes kind of randomly, and it's not, in experimental settings, it's not good. So this is a challenge to understanding precise and reproducible patterns of gene expression. When they say precise and reproducible, they mean like, if we're looking for like pattern formation, and we're looking at the expression of genes that lead to pattern formation, it's going to be harder to get that sort of sequence of events. It's going to be harder to see the effect if we have noise obscuring the process. And so we want to have an experimental setting where we can have a highly controlled process, where we don't have a lot of extrinsic noise, or noise that's coming from the outside, usually technical noise. Sometimes that extrinsic noise is biological, and so we want to minimize that. We only want the sort of noise that you see in the biology. Here we investigate this phenomena using gastruloids, a three-dimensional in vitro model for early mammalian development. So they're using gastruloids. So we talked about organoids before, and there are these different classes of organoid. You have gastruloids, assembloids, neural, neural organoids, things like that. And gastruloids are a class of them. So this is like kind of a model of gastrulation, if you want to be technical about it. It's a bunch of cells that you have that are sort of undergoing this gastrulation-like process. And so this, this model encompasses growth dynamics and gene expression patterns. We observe a remarkable degree of control over gene expression along the main body axis. So this is anterior to posterior. You should see reproducible sort of pattern formation from anterior to posterior. And in gastruloids, you also observe this control over gene expression with pattern boundaries positioned with single cell precision. So again, if you look at a normal embryo, you look at it like in vivo, you see these patterns forming. You see them from cell to cell, and you can see this in gastruloids. The idea behind gastruloids is we can produce this experimental test bed that reproduces the biology. We're not sure that we get every part of the process right, but if it's producing a certain output, and then we can measure that output, that looks very similar to mammalian embryos, then that's good enough, and that's what we're trying to do here. And so we have a remarkable degree of control over gene expression along the main body axis, with pattern boundaries positioned with single cell precision. Furthermore, as gastruloids grow, both their physical proportions and gene expression patterns scale proportionally of system size. So this is the other part about gastruloids, is that they're not just like precise with respect to some of these developmental processes, but they also grow. Embryos will grow in size as the embryonic stage progresses. And you should see this in gastruloids as well. In other words, you shouldn't just see like that, you get like these kind of immature systems that grow, and the processes of, maintaining pattern formation with growth. So as the more cells you get, you don't get this sort of diffusion. I don't know how else to put it, but diffusion of pattern formation. In other words, if I have a system with a small number of cells, I get these excellent sort of striping patterns. But if the embryo grows to like 3 times that size, the striping patterns will break down and you get this sort of smear. And that's not what we want. What we want is to maintain that striping pattern no matter what the size. And that requires, of course, for those new cells, those, because as we grow, we gain cells, we gain mass, to have that space and those cells express the same patterns, and ultimately to express their genes in that coordinated manner. So notably, these properties emerge spontaneously in self-organizing cell aggregates, distinct from many in vivo systems constrained by fixed boundary conditions. Our findings shed light on the intricacies of developmental precision, reproducibility, and size scaling within a mammalian embryo, suggesting that these phenomena might constitute fundamental features of multicellularity. So what they're doing here is they're trying to get a handle on sort of this multicellular self-organization. And, like I said, in C. elegans, embryogenesis is very precise. We see this in the model that Lalith was showing, that we can actually derive these higher order structures from embryogenesis in C. elegans precisely because it is very ordered and predictable. And, a lot of people, for the most part, C. elegans embryogenesis is what we call deterministic, which means that we don't have a lot of variation across organisms or across the process. You get cells where you know what fate its daughter cells will have, or you have these sublineages where you know that the ultimate fate will be of that cell lineage or sublineage. And so we don't have that in mammalian cells. What we have is sort of an activity dependence within the embryo. In other words, different cells are exposed to different signals in the embryo. And depending on those signals, they get that contributes to pattern formation. So it's a very different model. It's regulative development instead of what we call mosaic development. And we see mosaic development in Drosophila. We see it to a certain extent in Xenopus. We start to get regulative development. Different parts of the embryo are more and more regulative. And then in mammalian embryos, the entire embryo is regulative. And you end up with these, it's hard to actually get a lineage tree in mammalian embryos that is stable. In other words, if I take one embryo, I get one lineage tree. If I go to another embryo, I get another lineage tree. So let's say I've sampled a bunch of embryos from like a cow or a sheep or a mouse, and I can get those embryos and I can trace the lineage tree. And the lineage trees are always different. If I were able to track each cell and track their descendant cells, you would get these different lineage trees. They're totally different, but it's basically producing the same set of patterns, the same set of forms. And the reason for that is because it isn't the cells that are deterministic, it's the chemical signaling that basically sets itself up in the same way within the embryo, leading to these patterns. And so what they're doing here is they're using gastruloids to reproduce this environment where you can control the sort of the growth and the shape of the culture, and then you can use the same kind of system to produce a sort of embryo-like pattern formation. So that's really, this is really interesting work. Um, they kind of go into some of the details of what a gastruloid is here. So multicellular development entails the meticulous organization of cellular identities and body proportions in both spatial and temporal dimensions. So as the number of cells grows in an embryo, we want to maintain the spatial relationships between different parts of the body that are emerging. So when we have an anterior to posterior axis, which is front to back, we have a head that's emerging. We have like a brain and, the different parts of the head. We have, regions like the tail, and those things will later differentiate. And so, but we don't want to just have like a system where, or we don't want to have, but I'm anthropomorphizing that a bit. But you don't want to have like evolutionarily, you don't want to have a system that is kind of like this space is going to form a head, and then the head, the embryo scales in terms of size, and so then the head is kind of misshapen because it doesn't scale with the system. Now, in vivo biological systems, this problem is solved by coordinated gene expression and signaling between cells. But when you have something like a gastruloid, which is sort of like a collection of cells that you grow in culture, you need to be able to reproduce the system and have it work. And the thinking, of course, is that embryogenesis is self-organizing, meaning that it doesn't necessarily matter what the sort of arrangement of cells are necessarily. The process proceeds apace. Unless the organization of cells is really out of whack, then you don't get a viable embryo. So this is an important point, that we need to kind of reproduce this multicellular aspect where we have sort of this scalable system and this reproducible system. Now they're mimicking gastrulation in gastruloids. So these are gastruloids or organoids. They're these cell cultures where three-dimensional cell culture where you have these cells that form higher order structures. Gastrulation is a key event in the process of development. It's where the body plan and the subsequent establishment of asymmetric body axes occur. So you get this sort of some of the first sort of patterns or some of the first shape differences that form. So you have this sort of sphere before then, and then gastrulation gives you this sort of first asymmetric transformation of the shape. Coordinated gene expression during this stage leads to reproducible patterns between individuals despite the noisiness of the underlying molecular events of gene regulation. So this is important because, if you had a system where it wasn't reproducible between individuals, you would have individuals that were very different. So if you had, like, say, a species of fruit fly, you would have the fruit fly would lay eggs. And if you didn't have this kind of coordinated gene expression, you would have, during different stages of development, you would have fruit flies that looked all different in all different ways, hatching from those eggs. And when I mean all different ways, you would have fruit flies that had heads in the middle of their body and heads on the back of their body and tails in the middle of their body and tails in the front of their body. And it wouldn't, not very many of these eggs would be viable, but also they would just not have any sort of species identity. That's not what we see. And of course, species identity is important for it being able to behave in a natural environment, have a high fitness, and so forth. And so this is an important mechanism in development. It's worth noting that if you go to yeast, yeast actually exhibit some sort of transient multicellularity. They form these clumps of cells, and this is often an environmental thing. Right? The physics of the environment and other properties of the environment sort of allow for these multicellular aggregates to form. We've talked about them in other, like I think in Archaea as well, that this happens. But of course, these multicellular aggregates don't have these same properties of coordinated gene expression. That is, the multicellular aggregates have different shapes, they're not really regular, and if one of these multicellular aggregates buds off, the buds or the descendant clusters will look different. And it doesn't matter because they're not reproducing themselves in the conventional sense. They're just forming these functional clusters, these functional multicellular structures for different reasons. And so this is important because you're reproducing yourself. You want to have this sort of system. And so the challenge of translating transcriptional variability into precise and reproducible gene expression patterns has captivated research across the spectrum of animal models from nematodes to vertebrates. And then they cite papers showing these model systems. Developmental processes have been conceptualized as a sequence of steps aimed at mitigating and correcting errors in the face of molecular noise. So this is how people have thought of development, especially in model organisms, that each step in development is about error correction or it's about mitigating noise. And so you have this molecular noise, you have this variation that occurs across embryos, and the idea is in development that needs to be minimized in order to get embryos that are basically look the same or behave the same or reproduce that species form with enough fitness to propagate the species. And that's the idea. And so these mechanisms are kind of built into developmental systems. Can you reproduce this in a gastruloid is the question. So in vertebrates, mechanisms such as differential specification rates and cell sorting, and these are again, like ways in which cells differentiate and form different structures. And those mechanisms have been described as error correction strategies. So there are different ways in which there are higher order mechanisms in vertebrate embryos. And we can revisit these in another meeting, but this is just kind of a point of interest here in these references 13 and 14, where people are interpreting these as error correction strategies. So how does an embryo get around having this noise or being in this inherently noisy environment? And there are different mechanisms for doing that sort of work in the favor of order as opposed to chaos. However, in the context of the early fly embryo, the precision of macroscopic body plan features can be traced back to the precision of maternal inputs. So again, this is where, there's this maternal input if you have like an egg or you have a live embryo, you have these maternal inputs, and you don't necessarily have this in a gastruloid. The gastruloid is in culture. The cells that you get in a gastruloid have sort of a lineage, but they're separated from the environment in which they are. So when a mother lays eggs or when a mother has a live birth, there are inputs from the mother, placental inputs that are sort of important to this process. Exemplified with a morphogen gradient bicoid, this precision is transmitted at the single cell level along the major body axis to zygotic genes before gastrulation. Such precision in flies challenges the limits of molecular noise, suggesting its successive developmental stages may have evolved to minimize noise transmission at each step, both across evolutionary timescales and within the spatio-temporal boundaries of individual organisms. So an intriguing consequence of this precision is that the scaling of gene expression patterns relative to system size. So this is kind of one of the consequences is that you have, as the embryo grows, these gene expression patterns scale. So if you have an embryo that grows maybe 3 or 4 sizes, those patterns, maybe like striping, will retain themselves. They won't break down. The stripes will just get wider and the stripes will get larger instead of breaking down or like, becoming maybe like aliased in some way where you have stripes that are overlapping. Scaling observed in both invertebrates and vertebrates entails the preservation of body plan proportions among different individuals. During development, scaling manifests at various levels, encompassing morphogenetic movements, gene expression domains, and other phenomena, reflecting the intricate interplay of regulatory mechanisms. So again, we're dealing with gene expression, we're dealing with these gene regulatory networks, and there's this interplay because gene regulatory networks, for one thing, overlap, so you can get certain networks turned on at certain stages of development and they produce different outcomes. You also have cells that interact with one another, so cell-cell communication being what it is. Sometimes cells recruit other cells nearby to express a certain gene expression network or gene regulatory network. Other times you get gene regulatory networks in in different parts of the cell, maybe like the descendant cells of a certain mother cell to express this same gene regulatory network. And so you have these mechanisms for sort of these regulatory mechanisms. Sometimes they're coordinated, sometimes they're not, but they need to be coordinated for some of these larger scale morphogenetic phenomena. In contrast to organisms with well-defined developmental boundaries, such as flies, frogs, or worms, mammalian development relies on self-organization and continuous growth. So they're organizers in the embryo, but there isn't like this top-down uh, regulation where there's a signal that's it's instructing the cells sometimes, but it's not determining what the cells do. It's just giving them a signal that says, this is what we do in this region of the embryo. And then if the cell migrates to another region of the embryo, they might follow another instructive signal or set of instructive signals. And if you do experiments where you displace different parts of the embryo, you can get interesting results with respect to the embryo and embryogenesis. So this is something, especially in mammals, where you have a lot of these instructive signals and they dominate over sort of like the deterministic model of C. elegans. This is an important thing to think about. In contrast to organisms with well-defined developmental boundaries, such as flies, frogs, and worms, okay, we did that. Quantitative assessments of reproducibility, precision, and scaling in mammalian systems has been limited, prompting inquiries into whether the precision observed in flies is even necessary, as mammals rely on different developmental mechanisms. So it may be that, like what we see in Drosophila, which is this highly coordinated gene expression that produces things like striping patterns and body segments and things like that. What they're basically saying here is that maybe you don't need that kind of precision in mammals as opposed to flies for some reason. Mammals have different developmental mechanisms, and maybe we don't have to have this kind of system, or maybe it doesn't exist in mammals. And so there are different types of self-organizing systems. There's, of course, scaling in flatworms during regeneration. So in a flatworm, which is if you're familiar with Michael Levin's work on flatworms, he's done this work where you can take a flatworm And flatworms are interesting because any cell is what they call totipotent, which means a single cell, any single cell in a flatworm, you can take that cell, remove it, put it into its own culture, and it can grow an entirely new flatworm. This is not true of most other uh, developmental modes, but in flatworms, it's the case. And Michael Levin has done these experiments where you can ablate cells in the flatworm and you can observe massive regeneration. So he's done experiments where he's obliterated all but one cell in the flatworm, and then that one cell will grow a new flatworm. And the idea is that there's a self-organizing system based on bioelectricity and other things um, that reproduce that sort of structure for the flatworms, so that the cells that proliferate post ablation or post transplantation produces the shape of the flatworm. So this is the kind of self-organization and scaling that you can see in biological systems. Now, of course, with that being said, you go to different systems. You go to like mammals or flatworms or C. elegans or fruit flies or Drosophila or whatever, and you see different modes of development. Sometimes you see this totipotency. Sometimes you see pluripotency, where certain cells that descend from a mother cell have a range of fates. And sometimes you have this deterministic model in C. elegans, where the daughter cells have a very specific fate that they will default to. And so they get into progress in vitro models, and some of the attempts at looking at in vitro models have included gastruloids derived from mouse embryonic stem cells. And those kinds of in vitro models are promising in terms of giving us this virtual embryo or this pseudo-embryo. And so we see a lot of different types of properties of these gastruloids. They mimic critical events in mammalian gastrulation through self-organized patterning. For example, they break symmetry, so they exhibit these asymmetrical events. They elongate along an axis that resembles the most posterior part of the mouse's embryo's anterior posterior axis, and they can be cultivated in substantial quantities, rendering them conducive to quantitative approaches. So they don't replicate the entire embryo, but they replicate critical features of the embryo. They have the symmetry breaking and this axial organization. Those are the two main things that they're going to investigate in this paper. So in the interest of time, I'm not going to go through much more of the text, but I do want to go through some of the figures. This is an example here in figure 1 of this reproducible gastruloid growth. So we see in this in panel A, we have the gastruloid. We have it sort of reproducing this embryo. They show this inset here where they show the anterior to posterior organization of the gastruloid, and this recapitulates the anterior to posterior organization of the embryo itself, but it's not an embryo, it's a gastruloid, so it's going to look a little bit different. But basically it has the structure where you get this difference between one end and the other. And so you can see that here. And then this graph shows the length in micrometers and microns, and then against time. So over time, you get this length increase, and you get the scaling with the length increase of pattern formation. And so then you have this, I think this is volume in microns cubed, and then you have, I don't know what n is, 10 to the fourth. And so you get this, I guess it's a number of them. And then over time, you get this change here. And so this is also over time. This, I think, is the number of embryo or number of cells, and then the volume over the number of cells. And this shows like different increases. And then this is another derivative of, I guess, number of cells. And this is an increase over time. So just showing that there's a scalability in the embryo and different ways to measure it. So, okay, so I guess these notations of N and notations of V. So, you have variability, which is V, and then N is the number of gastruloids with identical age. And so it's age of the gastruloid and then the volume, or the variation, actually, not volume. So they did some investigations of these systems. Figure 2, this is where we see scaling of anterior to posterior gene expression pattern in gastruloids. So in embryos, we expect there to be this difference in gene expression between the anterior end and the posterior end. And remember, we had it kind of turned on its side, but here the anterior end is at the top, the posterior end is at the bottom of the image, and we're staining for these different genes and their expression. So we have SOX2, CDX2, BRA, FOXC1. So these are all important genes in development, mammalian development, and you see that there are these differences in terms of polarity. So you have certain gene, well, they're all like uh, broad CDX2 and SOX2 all have enhanced expression at the anterior end of the embryo in different ways. You can see this in the color in the heat map here. And then in the bottom, FOXC1, that's actually expressed posteriorly. And this is just kind of showing this over, I think, time. So this is where, we have this change and also in terms of size. So it's growing in size over time, and you get this expression. And you can see the expression gradient is increasing in size, even though it's the same proportion of the embryo. And so you can see that here as well. This one here is just showing the expression over this normalized area over time. So you have SOX2, CDX2, BRA, and FOXC1. And you can see that actually this is x over length. So this is the length of the embryo here. So this is anterior end here, 0.1, posterior and 0.9. So you can see that this is the length of the embryo. You have or actually, I think this is anterior, this is posterior. It's flipped around. So 0.9 is anterior, 0.01 is posterior. So you see the anterior for SOX, CDX2, and BRA, and then FOXC1, it's totally reversed. You have a different gene expression pattern. And then over time, there's a shift. You can see a little bit of a shift, but it maintains that sort of pattern. And then, of course, this is just kind of showing this sort of correlational analysis here, or regression analysis here for these data. Figure 3 just shows single pattern boundaries in gastruloid, precision in gastruloids. So again, we want to have precise boundaries. We don't want to just have, boundaries that are kind of loose. We want to really model the system and get the kind of precision we get in actual embryos. So here we have an example here of these different genes. This is the axis from 0.8 to 0.4. And then we have the variation here over length. So we can see that there's this variability that we see, but it's also quite precise in terms of sort of the region and where things are expressed. So you get some variation, but you also get this, tight regulation of spatial restriction. So you get like this green curve here, which is SOX2. You get this as opposed to this other curve, which is BRA, which is this yellow-orange color. And then Fox C1 is kind of in different, it's kind of showing up in different ways. So it varies by the gene, but you get these highly precise results, at least for some genes. And you don't get them all over the place usually. So that's the idea. Okay, so I think that's it for that paper. I wanted to go over that to show kind of the, this idea of these virtual embryos or these pseudo-embryos, and using gastruloids, how that can be achieved and how technically they're kind of can be very similar to embryos, mammalian embryos, which are actually harder to analyze than C. elegans embryos. And I'm not familiar with any artificial systems for C. elegans embryos, primarily because we don't really need them. C. elegans embryos are very easy to work with, relatively speaking, and so people just use those. So that's what I had for that paper. Did we have any questions or comments?
What was the title of the paper again?
It was precise and scalable self-organization of mammalian pseudo-embryos. This is in Nature Structural and Molecular Biology. I believe from 2024. So
Anyway, the differences are interesting between different types of embryos.
yeah, I think, and like, this is a system where it's just kind of a generalized mammalian embryo. So I don't mean to say that there are no differences between mammalian embryos. I mean, in terms of the mechanisms, they're similar. I mean, you don't see like this sort of thing you see in flatworms in mammals. You don't see that kind of totipotency. You see totipotency right at the beginning of embryogenesis from the one cell stage, but beyond that, it's all pluripotent to an extent. So you get this increasingly restrictive pluripotency, and then you get differentiated cells, which actually sometimes can be plastic even into adulthood. But that varies widely across mammals too. You don't just have
sorry, the dog. If you look at blood development, that's interesting. Erythropoiesis, or no, probably pronouncing that wrong. But anyway, if you look at that, the pluripotency in that is interesting.
Yeah, very interesting stuff. So we don't have any more comments. Why don't we call it a day for a meeting? And maybe we can follow up on this later. And thank you to Lalith for giving a presentation on the hypergraph stuff. I'm looking forward to seeing the paper. Okay, thank you. Have a good week. So in light of the paper we did today, I'd like to talk about two other papers. This one is from Lucy Ham, Michael Stumpf, and colleagues. This is, I think it's from Nature, and it's called a stochastic versus deterministic perspective on the timing of cellular events. So we talked about sort of stochasticity versus determinism in embryogenesis before. And there's this whole literature on how we can model developmental cells or cells in embryonic development as either being stochastic or deterministic. So in C. elegans, we have a deterministic model where a cell has a lineage or sublineage that it belongs to, and depending on that lineage, which is the history from mother to daughter cell, you have this switch that happens, and those cells can only have a certain defined set of fates. So you have, say, like a sublineage where you have a mother cell, and that mother cell proliferates into daughter cells, and then more daughter cells, and it might produce something like muscle. And those cells their fate can't change suddenly in the middle of a lineage tree, even with external perturbation that produces a gene expression noise. Usually that noise is buffered to attain that deterministic fate. But there are also cell types where there's a stochastic aspect to cell fate. And so in a lot of cellular reprogramming experiments, for example, or in mammalian embryos more generally, there is this stochastic sort of cell differentiation, meaning that you can have different cell fates. A cell can be pluripotent or even totipotent, but the determination of that is not switch-like, but it's this sort of random process by which, depending on the signaling that occurs in the cell or the genes that are expressed, it produces a certain outcome. So there's this model of stochasticity versus deterministic cell differentiation, and people have applied mathematical models to it. So that's what they're referring to here. So if we read the abstract of this paper, cells are the fundamental units of life, and like all life forms, they change over time. Changes in cell state are driven by molecular processes. Of these, many are initiated when molecule numbers, or the stoichiometry of a cell, reaches and exceeds a certain threshold. So there's a stoichiometry threshold, meaning that if you have, say, a certain copy number of mRNA molecules of a certain gene that's expressed, you can hit this threshold and it changes this, maybe it changes the state of the cell. There's certain genes in mammalian cells that are sort of the key pluripotency cells, and if you overexpress them, you can induce pluripotency in a cell. Things like this. A characteristic that can be described as a digital cellular logic. So they're proposing that the whole area of reprogramming, or the whole area of sort of stochastic gene expression, can form this digital cellular logic. Here we show how molecular and cellular noise profoundly influence the time to cross a critical threshold, first passage time, and map out scenarios in which stochastic dynamics result in shorter or longer average first passage times compared to noiseless dynamics. We illustrate the dependence of mean first passage time on noise for a set of exemplar models of gene expression, autoregulatory feedback control, and enzyme-mediated catalysis. Our theory provides intuitive insight into the origin of these effects and underscores two important insights. So the first is that deterministic predictions for cellular event timing can be highly inaccurate when molecule numbers are within the range known for many cells. And then two, molecular noise can significantly shift mean first passage times, particularly with an autoregulatory genetic feedback circuits. So, this is kind of speaking to this idea of using stochiometry as a switch to determine fate. So if I have a certain gene that maybe is a key gene in determining pluripotency, or determining cell fate, or determining maybe the ability to switch a cell fate, I have to have a certain amount of that in the cell. And so this is, of course, has to be at a threshold of some type. So the accumulation of this molecule has to be, it's not an even process. Sometimes if the process of gene expression is uniform, you can reach that threshold within a certain amount of time. But of course, there are recycling dynamics. The molecules degrade, the molecules are produced, and sometimes they're bound by other things in the cell. And so the amount of that molecule is going to, the sort of the net amount of that molecule is going to change. And so you have this time to threshold that can vary across cells, especially when you're talking about noise in gene expression, like we talked about in the last paper, where gene expression isn't this sort of constant pulse of production, but it's cyclical, and it can be stochastic in the sense that it's random in that cyclical nature. So it can pulse at different scales. So this is the kind of thing they're thinking about here. This first point here, deterministic predictions for cellular event timing can be highly inaccurate. So if we use a deterministic model to say that if we have a certain population of cells and we overexpress a gene, that those cells will all become a certain type of cell or all become pluripotent, you can't assume a deterministic model for that. You have to assume that not only do cells have to reach this threshold, the stochiometric threshold, but that they're different in every cell. And so, I remember when I used to work in cellular reprogramming, we spent a lot of time thinking about this problem. It seems like a very trivial problem. But if you introduce genes into a cell and you overexpress certain genes, what's the time to sort of like a threshold where those cells can become pluripotent? And it was not an easy task, because it's not like you just introduce the gene into each cell and then those cells will become pluripotent after a certain amount of time. Some cells will become pluripotent quickly, some cells will become pluripotent later, and some will never become pluripotent because the process underlying it is stochastic. The second point here is that molecular noise can significantly shift mean first passage times particularly within autoregulatory genetic feedback circuits. So molecular noise plays a large role in some of this variation, but also when you have gene circuits that are autoregulatory, meaning that they are regulating themselves, especially in the presence of some signal that pushes in one direction or another. So this is all to say that cellular dynamics are not deterministic. Observation of RNA or protein number in single cells show that variation with time is stochastic. And so they get into the idea of expression noise and biomolecular noise and how that can affect copy number and stoichiometry within the cell. So we can use the chemical master equation to model probabilistic descriptions of chemical reaction kinetics. And this has helped us understand some of these mean behaviors of cells. But the mean behaviors of cells are just that. They're mean behaviors. They don't represent every cell all the time. There's a lot of variation around that mean, and that's what we kind of want to understand. And so this is where this whole area comes from, about thinking about the stochasticity of these processes and how they deviate from the chemical master equation, and how we can get, we can at least appreciate that. I don't know if we can really find a different predictive model that will help us, because stochastic models generally aren't predictive, and that's the whole point. They're stochastic. So this gives a little example from cell cycle dynamics. So for example, in cellular development and differentiation, progression through different phases of the cell cycle only occur when certain cyclic proteins surpass a prescribed threshold. Many physiological processes are also triggered by a certain threshold. And so we need to have sort of these thresholds, and we need to figure out what the dynamics are leading up to these thresholds. And that's kind of the best we can do in terms of predicting this from cell to cell. So this is an example here, first passage time, which is to say that, you have this, once the amount of this molecule hits the threshold, it triggers some event, and then the amount of this protein or mRNA concentration can fall below the threshold, and that event has been triggered, and it doesn't matter after that. It just needs to hit the threshold once. And the question is how fast can it hit the threshold given these stochastic dynamics? So this is where we have protein number on the y-axis, time on the x-axis. And protein number builds over time, and it's fluctuating. And if it hits the threshold, it triggers this apoptotic event. If it never reaches the threshold, which is possible, it goes into cell cycle arrest. Sometimes there's a time limit on this process. So it's time limiting in the sense that if this concentration never reaches the threshold before it reaches a certain time limit, then it'll by default go into cell cycle arrest. And if it hits the threshold before that time, then it goes into apoptosis. But once it hits the threshold, it makes a decision about fate. Sometimes it's time limited, sometimes it's not. This example here in B shows that there's this very high degree of variation in the first passage time. So like we have protein number versus time. So we're dealing with P53, which is a protein. And we can use any protein, and there are different profiles for this. But we have a number of different cells where P53 is expressed, and you see that it forms this distribution around the first passage time. In other words, there are different times when it hits the first passage time because of the stochastic dynamics. But you can actually look at that variation and plot out a Gaussian curve, which describes sort of the mean time to first passage, and then some variation on the other end. So when you have you're talking about variability and noise, you're talking about this stoichiometry here, this amount of molecular, the number of molecules in a sample or in a cell, but also its time to first passage, which is also variable. So we have this process. We have this gene that's being expressed. We have these kinetics to produce proteins of different sizes. There's this burstiness in protein production. And then we have this target number of degraded proteins, which is this, we have to basically wait a certain amount of time to get a certain number of proteins. And so this is the model here. And so we have, in this case, we have protein number over time. So we have our protein number, and we're looking at variation. We have these stochastic dynamics. We have protein number over time here, which is showing this. We have the we have sort of the power law dynamics here, where we have eta over log k, and then we have this other set of bursty dynamics characterized by this power law, eta over log k, where we can see that it's not a linear process, it's actually a nonlinear process. So one of the core parts of their model here is this bursty birth-death process. So we consider the bursty birth-death process with fixed burst size R. So a birth-death process is where you look at a pool of some molecule or cells or organisms, and the number of constituents in that group is governed by two things: births and deaths. So those constituents have to be born at some point, and then they have to persist. And as they persist, they're still there. As constituents are born, that adds to the number of constituents. But then some of those constituents die and they're removed from the pool. So the birth-death process governs the size of a set of constituents in a population of cells or individuals or molecules. And so that process can be bursty, meaning it's not linear. So you think about births and deaths, like of humans in the hospital, where there's certain periods where a lot of babies are born and other periods where there are no babies being born, and that's normal. You also have that with cells, where there are a lot of cells being born at some points and no cells being born at other points. If you look at, you do a census at any given time, you can see that birth rates are bursty, but also death rates are bursty. Sometimes there's a lot of apoptosis in a cell. Sometimes there's very little apoptosis in a cell. Sometimes, or in an embryo, sometimes the same thing happens with necrosis. These can be natural fluctuations, or they can be due to some stimulus on the outside. We consider the bursty birth-death process with fixed burst size R, so they fix the burst size, and they show that basically this is the model here. It is assumed that there is a continuous production of a molecular species P in bursts of fixed size R, according to a Poisson process at constant rate K. So a Poisson process is modeling arrivals, and it's nonlinear. It's an exponential distribution, and then there's this rate K of the Poisson process, which is constant. The degradation process captures both active degradation as well as dilution due to cell division, and is modeled as a first-order Poisson process with a rate of that. Throughout the remainder of the paper, we scale time as tau equals your variation times time, and assume all model parameters have been scaled by the degradation rate, so that equals 1. Hence, both time and rate parameters are non-dimensional. The bursty birth-death process serves as a crude model for bursty expression of either RNA or protein due to transcription or translation. We do not here account for the fact that burst size in these cases is not fixed, but is distributed according to a geometric distribution. So they go from a simple birth-death process to explain that, then to this bursty case, and then thinking about different burst sizes. So the burst size is this parameter R, and it can be 0, or it can be greater than 1. And so then that determines sort of the variation in our process, and how long it takes us to reach that first passage. And so to model bursty gene expression, we can use something called the telegraph model. This is where we want to know whether or not the same transition in the ratio of the deterministic to stochastic waiting time from below to above 1 is possible. And so there are these effective reactions here. In the model, the gene switches probabilistically between the inactive state G star and active state G, from which mRNA molecules, or M, are synthesized degradation of mRNA occurs independently of the promoter state. So you have this switch between G star and G, an inactive to active state, and that determines how mRNA molecules are synthesized, and then the degradation rate may be constant, and that's different from the production of mRNA. So your pool is, it fluctuates stochastically because you have a parameter that's controlled by the synthesis rate, but then you have a different process, the death process, which is a different set of dynamics, and that leads to this sort of burstiness in the population. Then there's this autoregulatory feedback loop in which a protein produced by a gene either enhances or suppresses its own expression. We see this commonly in biology. We see this in E. coli, where we have negative autoregulation for around 40% of known transcription factors. A number of studies have used stochastic models to investigate optimal feedback mechanisms to minimize the variability. The reaction scheme for the feedback loop we employ is given here. This shows this where G star and G represent the bound and unbound states respectively, and P represents protein. So you have these other parameters that involve the unbinding and binding rates, and then the variation is the protein degradation rate. And so this is just the feedback loop described by these parameters. And it describes sort of the fluctuations in this process where you have births, deaths, and then how those are regulated. And so we measure this by looking at the steady state mean. So again, the steady state mean helps us characterize this sort of process, but it by no means describes the full reach of the process. So they talk a little bit about the difference between deterministic and stochastic model prediction. So for simple birth-death processes, the deterministic model prediction provides a strict upper bound predicted by the stochastic model. In other words, intrinsic noise leads to a reduction of the trigger time. This effect becomes more appreciable as the mean steady-state molecule number decreases. For the bursty birth-death process, there is a switch from the regime where intrinsic noise reduces the mean trigger time to one where it lengthens it as the target molecular value is decreased. So there's this relationship between trigger time, between mean passage time, and then between those, and then the size of the population of molecules, or the stoichiometry. Since the expression of many genes is bursty, of transcriptional or translational origin, the simple model suggests that the transition may exist in more realistic models of gene expression. Indeed, we showed that the same transition occurs in the telegraph model. And so this brings us to autoregulatory feedback. So for feedback loops, we observe significantly higher ratios of deterministic to stochastic FPTs compared to previous models. This has implications in the timing of developmental processes governed by molecular clocks, where dynamics are often modeled deterministically. So these are things like circadian clocks in Drosophila and in Neurospora, or segmentation clocks in vertebrates. Additional error results provide insights into the non-monotonic dependence observed in both the deterministic FPT and MFPT on the feedback strength. And this is non-monotonic behavior that has been shown in stochastic threshold models. The effect of feedback on the differences between the predictions of the deterministic and stochastic frameworks are well known to increase with decreasing system size or volume of the system, and with increasing proximity to the point in parameter space where a system switches from stable to unstable dynamical behavior. So in a lot of stochastic systems, predictability is hard because when the stoichiometry is small, when the number of molecules is small, there's a large amount of variation. Variation can be amplified by the law of small numbers and that sort of thing. So when you have small numbers of molecules, the variation can be much higher. When you're dealing with larger numbers of molecules or larger pools of molecules, differences, stochastic fluctuations of less have an effect, and so it's easier to predict. So at the end here, they say, our study has shown that intrinsic noise has non-trivial effects on the timing of cellular events, and that simple models can provide intuitive insights into the microscopic origins of these effects. The hope, whether implicit or explicit, that deterministic models predict accurate mean quantities appears misplaced for the MFPTs, which have important implications for modeling-based studies of biochemical timing. So as a note here, FPT is first passage time, and MFPT is mean first passage time. So when we use FPT and MFPT, that's what we're referring to.